This lesson is on the first part of section 5.2, passive transport. So the things that we're going to cover here for how cells move things in and out of the cell, we're going to see diffusion, osmosis, facilitated diffusion, and diffusion through ion channels in this section. So the simplest type of transport is just simple diffusion. This is the movement of molecules from an area of higher concentration to an area of lower concentration. All right, and so how you're going to see this written from time to time is with brackets around it. If you see brackets around something, that just means the word concentration, how concentrated something is. Okay, and hopefully you got notes on that from yesterday. But those brackets mean concentration. Essentially, if something is higher, it's got more stuff dissolved in it. It's got more solutes dissolved in the solvent. Okay, if the concentration is high. If the concentration is low, it has less stuff dissolved in it. It's kind of the difference between Gatorade and then watered down Gatorade. But for simple diffusion, the movement is always from areas of high concentration to low. So from where there's a lot of particles dissolved in water to where there's not as much dissolved in water. The difference in concentration of molecules is called a concentration gradient. So let's say I had like a membrane here, and on one side I had a bunch of particles, and on this side I only had one. Okay, this is called a concentration gradient. There's more on one side of the membrane than there is on that side. And for simple diffusion, what's going to want to happen is some of these particles are going to want to move over here to even out this concentration. It's moving from high to low. It's driven by kinetic energy, which is the energy of motion. Molecules tend to spread out evenly. That's what they want to do. So they're going to move from that more concentrated to less concentrated side. Okay, so that's what they're going to want to do. They move what we call down their concentration gradient. Okay, so this is the higher concentrated side over here. Okay, where that arrow is. This is the lower concentrated side, so we say it's moving down its concentration gradient from the high to the low. If there's no other influence acting on this, so the cell's not actively trying to change something about it, eventually this reaches equilibrium, where the amount going in this direction to the right is equal to the amount going back to the left, and we kind of reach this point called equilibrium. Okay, so here's just an example from your textbook. All right, so let's say you had sugar molecules down here in the bottom, a sugar cube, and then you filled it with water. Over time, what happens? Well, as this begins to dissolve, as the sugar begins to dissolve at the beginning, it's more concentrated down here at the bottom. Okay, so the top part of this water would not be as sugary, but over time, as this thing dissolves completely, the particles tend to spread out and evenly distribute in that water. Okay, that is just simple diffusion. Right, that's what this is an example of here, simple diffusion. Okay, so what can get through the plasma membrane? Cells allow some substances to pass through. Obviously, they can't let through everything, right? That plasma membrane is there to protect it from allowing just anything through. If the substance can pass through, then diffusion from high concentrations to low concentrations is what's going to happen, right? It has to move from where it's highly concentrated to where it's low concentrated. Okay, so if it can, to begin with, it has to be able to go through. It's moving from high to low. Diffusion across the membrane is called simple diffusion and allows only certain molecules. It depends on size and type of molecules. Some stuff, okay, I'm going to rewrite this down here, it can pass through readily. Okay, and that's because of its size and its polarity. Small things like CO2 and O2 can pass through the phospholipid bilayer. These are really tiny molecules. Okay, so they actually can go through that bilayer without being transported in by a protein somehow. Okay, that's why. It's because they are small. So how do we get in bigger stuff? Like how do we get in like sugars and other carbohydrates, right? How do we get that stuff through? That's going to take some effort on the part of the cell. Okay, so osmosis, you may have heard of that term before. This is just going to be diffusion of water. Now, it's a little bit backwards in terms of like thinking about it, but it should make sense. It's usually, right, water is usually the solvent in solutions, right? It's the stuff that dissolves things. So osmosis is the process by which water molecules diffuse across a cell membrane from an area of higher concentration to an area of lower concentration. Does this require energy, right? Because it's going from higher to lower concentration, the cell does not expend energy. In fact, and I should say this more specifically here, and state clearly, passive transport and everything in this video, okay, passive transport does not require energy, meaning this happens naturally, okay? The other video, active transport, the cell is going to need to use energy to transport stuff. Here, these are particles that are going through without the cell expending energy, okay? This is what passive transport is. So we've talked about simple diffusion, that's one, and osmosis is another type, okay? So this is what passive transport is. Which way does the water flow? Well, it depends on the concentrations of solutes on the two sides of the membrane. All right, we'll see an example of that here in just a sec. Okay, we have some terms here: hypotonic, hypertonic, and isotonic. Okay, so let's say we have a membrane here, okay, that is impermeable to solutes, okay, but it is permeable for water. Okay, so let's say we can let water come through here, but we can't let solutes go through here. So we'll say that this is the inside of the cell, this is the outside of the cell. And what sort of situation are we going to get here? Well, for hypotonic, the concentration outside of the cell is less than the concentration inside. So we'll just put one particle there and 3 particles here, okay? All right, so the concentration is greater here, and it's less on this side. I can't move the particles across this time like I could with simple diffusion, but I can move water. And what I want to do is try to even out the concentrations. Right now it's higher concentrated in here. Well, I can lower this concentration by adding more water, by taking water from this side and moving it over here. So the net result in this situation is we are going to have water move this way. Okay, it's going to go into the cell to help dilute down this side. Okay, it helps to dilute it down. Remember, we can't move the particles across the membrane in this situation. Okay, the next example. All right, we'll draw another membrane here. Again, we can't move particles across, but we can move water. So in this case, it's higher concentrated outside of the cell than it is on the inside. If I could spell outside, there we go. And there we go. On the inside, I can't move the particles, but I can move water. Water is going to want to try to dilute down the higher concentrated side. So what's going to happen is the water is going to move out of the cell, okay, and to help dilute that down. All right. We could have a situation where we have something called isotonic. And that's where our concentrations inside of the cell, oops, I don't know why it changed color here. Oh, there we go. The concentrations inside of the cell and the outside of the cell are going to be the same. Well, it's not that water doesn't move anymore, it's just that water moves in the forward direction and the reverse direction at the same rate. Okay, so it doesn't change. So kind of what happens here, well, if you can imagine water moving into the cell, right, and trying to dilute this down, that's going to cause the cell to get bigger as all this water rushes in to try to dilute down some of this solute. Okay, that's going to make the cell get bigger, right? Cell gets bigger. If you have a hypertonic solution where the outside is more concentrated, the cell is going to shrink, okay? And that's because the water is going to leave the cell. Here it's going to remain the same size, okay? Now remember, these are all relative. Water diffuses from hypotonic to hypertonic solutions, all right? Hypo to hyper. Okay, so here's an example with red blood cells. And as you can see here, right, Normal situation, external solution is hypotonic to the stuff in the middle there, the cytosol, so water is going to move into the cell. If you have an external solution that is hypertonic to cytosol, meaning there's more stuff on the outside, the water is going to move out of the cell. Okay, so if you want to think of it like that, here, let me change the color of that. Here we have more particles inside of the cell. Here there are more particles outside of the cell. Here we're seeing equal amounts on each side, no net change. Okay. So how do cells use this? Cells in isotonic external environment keep water moving in and out of a cell. So things like vertebrates on land or organisms in the sea. Cells in hypotonic solution always have water moving in. So these are like freshwater organisms. They must rid themselves of excess water. Okay, so things that live, say, in these hypotonic solutions are constantly having water trying to get into the cell. So they have to actively try to move that out. Okay, so that when they remove it, it's going against a gradient. It's going up the gradient instead of down, and that's going to take energy. So that's going to be for the next section. Okay. All right. Here's an example, something called plasmolysis in hypertonic solutions. Essentially what ends up happening is, let's say you put a plant cell, okay, in normal hypotonic solutions, where the solution is hypotonic, the water is going to rush into the cells, which is going to fill those vacuoles, right, the central vacuoles, which is going to cause these cells to have pressure against their walls. They're going to expand. It's going to keep the plant upright and keep it from wilting. If you take that same set of cells and now you have a solution where there are more particles outside of the cell, right? So say you put it in like salt water and it's not normally a salt water plant, okay? What's going to happen is the water is going to leave the cell and the cell is going to lose the pressure. The cell walls are going to shrink. Okay, the cells start to shrivel, and the plant starts to wilt. Okay, so this actually is a big thing here, right? If you put like a plant in like just normal water, okay, that water is going to go in and fill those cells because there's more stuff inside of the cell than there is outside. But if you put it in like salt water where it's got more solute outside of the cell, the water is going to leave the cell. Okay. Okay, going back to red blood cells for a second. What happens if a cell cannot adjust to the environment? So what if, like, plant cells are pretty resilient. They have cell walls. They can prevent themselves from expanding too much. What happens if a cell can't do that, right? So for instance, like red blood cells. They don't have any way of ridding themselves of excess water. They don't have these, like, that they're called contractile vacuoles, which pumps water back out of the cell, right? Okay, they don't have that. They don't have pumps. They don't have cell walls. So what happens? Okay, well, we already had seen an example of what happens if you put it into hypertonic solution. The water leaves the cell and the cell shrivels up. The other side of things is, what if you put, say, red blood cells in just pure water? Well, now the concentration is greater inside of the cell, all right, and water is going to rush into that cell. But there's no cell wall to prevent this thing from expanding too much, right? It's just going to keep filling with water. And eventually, it's going to burst, okay? This is called cytolysis, is what that is. So you can actually kill some of these cells that don't have ways of mitigating this excess water coming in, okay? If it just keeps filling, eventually these will burst. Okay, so facilitated diffusion. What about large molecules? Okay, so we talked about how things like CO2 and O2 can diffuse in freely. We talked about water with osmosis. So how do we do with the rest of this? Well, large stuff has to come through, right, by some sort of mechanism. So facilitated diffusion is used for molecules that cannot readily diffuse through membranes, even when there is a concentration gradient. So this is big stuff, stuff that's so large that even if it would make sense to go from a high concentration to a low concentration, it's not going to work. So what do? Well, it's got to be assisted by specific proteins in the membranes, and these are called carrier proteins. So carrier proteins are going to facilitate diffusion. Okay? It's still moving from high to low concentrations. Therefore, it's still passive, right? It doesn't require energy. We're still going from high to low. All right, but this time it has to be moved through by a protein, okay? It can't just go through the membrane. So an example here is something like glucose, right? This is a carbohydrate that's going to provide energy. It's got to get into the cell somehow or another, but it's too big to just diffuse through. So instead, we're going to have these carrier proteins that are going to help get glucose into the cell when the concentration is low, right? So it's going to be lower inside of the cell than it is outside of the cell, and it's going to get put in there. So here's an example of facilitated diffusion. So here's our plasma membrane. Here's a carrier protein. All right, right now, there's a higher concentration of particles outside of the cell than there is inside of the cell. So we're trying to get some of these into the cell. They're too big to go directly through the membrane, so they have to go through this carrier protein. So step one, molecule binds to a specific carrier protein, and the carrier protein changes shape. So when it comes here, it's going to change shape. Step two, the new shape yields a molecule from hydrophobic interactions of the bilayer, so we're going to get sort of this sort of shape, and then it's going to get pumped through here. So step three, once shielded, it's now in between all of this. It's got its own pathway through. It doesn't have to go through the bilayer. The molecule can be transported through the cell membrane, which is what's happening in step three. In step four, on the other side, the molecule is released and the carrier protein returns to its normal shape. Okay, and this would continue to pump stuff through, okay, until you reach that equilibrium point, right? At which point the concentration inside of the cell was the same as the concentration that is outside of the cell. Okay, so those run without having to expend any energy. So a quick summary for facilitated diffusion. It is the proteins, I should say here, are specific to one molecule. Okay, they only fit one thing. So back in that picture there, that protein that was moving stuff through, that one was only for that particle. It's specific to one molecule, which means if you want to move glucose in, you need like a glucose carrier protein. Okay, if you need to move some other type of carbohydrate in, you need a different protein to do that, but they're all embedded in that plasma membrane. Number two, it helps large molecules move into or out of the cell. Okay, so unlike simple diffusion or osmosis, this is going to help large stuff get through. And number three here, right, this is still going to be passive transport, so it moves from high concentrations to low concentrations. So that's still going to happen. Okay, diffusion through ion channels. What about charged atoms or molecules? Things with charges, plus or minus charges, trying to get through plasma membranes, it's difficult, okay? They're going to need some help, right? They can't just diffuse through this. So how are we going to get these charged particles through the membrane? We have something called ion channels, all right? So we have to have some sort of thing in here, okay, something in the plasma membrane to help this move through. We're still moving from high to low concentrations, which means this does not take energy, not take energy, okay? It's still passive. An example here, a sodium channel, a calcium channel, a chloride channel, they're all able to move, okay, charges, ions through the plasma membrane. Now, even though ions are small, their charges prevent them from diffusing through the plasma membrane. So before, when we were talking about facilitated diffusion, with molecules like glucose, C6H12O6, okay, we needed a carrier protein to get them through. Here we need an ion channel to move this through, even though these things are a single atom. So something like a sodium here, okay, this is going to need an ion channel. Even though the sodium is small, right, that large charge, the charge that's on it is going to prevent it from just naturally moving through. That C6H12O6 is so large, the diffusion is going to have to be done by carrier proteins, right? Okay. Diffusion by carrier proteins. Channels are specific for one type of ion, so just like what we were talking about just before here, facilitated diffusion, those channels are specific for one ion. So the one that I say drawn up here, okay, is only going to work for that particular negatively charged ion. I would need some other sort of, I don't know, shaped protein here. I'm just making up an example for the positively charged one to go through. Okay, we need a different protein. Some are always open, others have gates. Those gates are controlled by cell membrane stretching, electrical signals, chemicals inside of cell or outside of the cell. But essentially, we always have to remember that it's going to be passive. It doesn't take any energy. It's still going down the concentration gradient.